So this morning I'll be talking about the radiobiology of hypofractionation. Much of my interest, both clinically and in the research area, has been focused on biologically guided radiotherapy. So how can we develop systematic methods that really allow us to derive prescription doses that integrate patient-specific information about tumor and normal tissue biology? And really this is a different dimension that we can optimize our treatments based on. And in the context of hypofractionation, I think there's a few big controversial questions that still sort of remain unanswered. The first is, to what extent does our classical understanding of radiobiology apply at high doses? So by classical understanding, I mean really the 4 R's of radiobiology that we've all learned. Specifically, I'll talk a little bit about DNA repair, tumor hypoxia, and reoxygenation, as well as the others. There's also been a lot of very interesting work around possible secondary mechanisms of biological response. So might there be a fundamental difference in biology between conventional and stereotactic radiotherapy? Of course, up until about 15 years ago, the primary mechanism of cell death was believed to be mitotic cell death related to the biological processing of double-strand breaks, but there may be other relevant secondary mechanisms when we go to really high doses. So I'll briefly mention those. And the other big question that also has some clinical relevance is, are conventional dose response models that we're used to using, are they valid at high doses per fraction? So I think we all know why this is such why these are very interesting questions. The utilization of stereotactic radiotherapy has been rising rapidly in recent years. This is just an example of primary early stage non-small cell lung cancer patients undergoing SBRT, and it's really becoming more mainstream clinical usage. And of course, this is because we're observing high local control rates. So now there's prospective clinical data now showing local control rates for early stage non-small cell lung cancer patients undergoing SBRT upwards of 90%. And we can compare that to conventional results that may have been as poor as 40% local control rates. So why are the results so good? There's been a lot of discussion around potential unique biological mechanisms. Zvi Fuchs and Rich Kolesnik at Sloan Kettering have done a lot of work in the laboratory looking at endothelial cell apoptosis and rapid tumor vasculature shutdown that could lead to secondary effect. Chang Song at University of Minnesota has done a lot of work on vascular damage producing secondary cell killing. And of course, there's been a lot of work focused on enhanced anti-tumor immunity after high-dose radiation. And I don't have time to go into the details of these mechanisms today, but I will point you to a recent Red Journal review that sort of looks at a detailed analysis of the evidence for and against some of these techniques. And I'll get back to this a little bit at the end when we're looking at some clinical data. So can we observe any of this in clinical data, which is, I think, the more relevant question. We must also remember the physics techniques. So what we're doing on the treatment planning and delivery side is very different. So we can talk about hypofractionation, but we also have to look at the radiotherapy techniques. So in conventional radiotherapy, we delivered parallel opposed or 4-field box fields with uniform prescription doses. We had a lot of normal tissue being irradiated with higher doses. And really moving towards stereotactic radiotherapy, it's been a paradigm shift. So we are limiting the normal tissue volume that's irradiating. We're allowing hot spots to be much larger in the center of our tumors. And this is really facilitated by physics advancements in the context of IMRT, IGRT, volumetric modulated arc therapy. And this is really letting us go to much higher doses. We have these large dose gradients. We can deliver higher doses, higher biologically effective doses in this SBRT paradigm. So that's another important thing to remember. How do we move towards hypofractionation? Well, you could say we usually deliver 39 fractions of 2 gray, say for a conventional fractionated prostate radiotherapy treatment. What dose per fraction would we have to deliver if we wanted to go down to 10 fractions or 5 fractions? Well, we could sort of do a trial and error approach, or we can guide our decisions based on dose response models. And this is the simplest kind of way that we would do that. I think you're all familiar with the BED calculation. It basically corrects for dose per fraction, and tissue radiosensitivity. Well, you can basically calculate the dose per fraction that you would need for a given number of fractions for a particular ISO effect, for the same biological effect. So here's a simple calculation for fraction size as a function of number of fractions for a low alpha beta tumor. And we might expect if this was prostate, we have 39 fractions of 2 gray. Then we sort of move towards moderate hypofractionation and to extreme hypofractionation. And these are actually, these vertical lines are actual clinical trials for prostate hypofractionation, with the most extreme being originally Chris King at Stanford, and then UCLA was doing his prostate CyberKnife protocol. So you can see they fall on this ISO effect line. And another thing I like to point out when we do these types of simple calculations, that the uncertainty in our assumed radiosensitivity, the further we get away from our conventional approach, the larger impact on the derived prescription dose it has. So that uncertainty in the biological parameters translates into an uncertainty in our derived prescription doses. And of course, we know that human cancer populations is quite heterogeneous. So we have not just a single unique radiosensitivity, but a distribution of values. The other thing these simple calculations neglect is our classical 4 R's of radiobiology. So what other biological factors that can impact our treatment effectiveness? So here on the y-axis, we have treatment effectiveness as a function of treatment duration. We know DNA repair will reduce our treatment effectiveness the longer we protract our dose. So if we deliver it in 5 minutes versus 2 hours, there will be a difference in biological effect. We know that reoxygenation and redistribution will work in the other direction. So if you fractionate over many fractions, reoxygenation of your hypoxic tumor will lead to an increase in treatment effectiveness. And of course, over a longer period of time, repopulation will diminish biological effectiveness. So these are other biological factors that we have to consider with these simple calculations. So let's talk a little bit about a couple of these. So DNA repair, we know that cell killing decreases with decreasing dose rate for the same given dose. If you reduce your dose rate, you see that corresponding decrease in biological effectiveness. This is quite well understood in vitro. We can actually model this. You can include DNA repair in the classical linear quadratic model. It modifies the beta component, which is the 2-track or dose rate dependent mechanism of cell death. And you can explain the in vitro data very well using a unique set of 3 parameters where you have the double strand break for repair halftime here. So what does this actually mean biologically? Well, we have repair of DNA damage that's occurring between and during fractions. So within a treatment delivery, you're having intra-fraction DNA repair, and that effect increases with increased delivery time. So if you're delivering over a longer period of time, that will reduce biological effectiveness in the context of SRS and SBRT. What about tumor hypoxia at high doses? Does hypoxia become irrelevant? Well, in vitro, the evidence is quite clear. You can go to very high doses. This is a colony survival assay curve for V79 cells. There's many published studies with different cell lines. Going up to even 30 gray, we still observe an oxygen enhancement ratio of about 3. And the OER values for cell death are relatively constant over a large dose range. So the problem of hypoxia doesn't go away at high doses in vitro here. So we actually tried to model this clinically. So how can you translate this to a clinical scenario? And I'll take a minute just to explain this plot because it's a bit complex. On the left side, we have a high alpha beta tumor. On the right side, a low. And basically, this is the log cell kill for a fractionated radiotherapy regimen. So under fully oxygenated conditions, we get about 8 to 9 logs of cell kill. And this is for a given number of fractions and the equivalent dose per fraction to yield BED under those normoxic conditions. So for example, with that prostate case that I went through previously, 39 fractions of 2 gray is here, 5 fractions of 7.5 gray is here. If our tumor is well oxygenated, that would be the ISO effect for fractionation. But what if we tried to incorporate hypoxia into the cell killing model? We can have a much more complicated model here. We actually see a trend towards reduced biological effectiveness in the context of hypoxia as we move towards hypofractionation. And that's sort of consistent with our classical understanding of how reoxygenation would work clinically. So this led to a clinical study at Yale that's currently underway, a pilot study that's IRB approved, where we're basically looking at performing serial F-MISO PET imaging in early-stage non-small cell lung cancer patients undergoing SBRT. So with the goal to look at how do these spatial and temporal distributions of hypoxia change in SBRT patients surrounding a single 18 gray fraction here. So this is our first patient that made it through the imaging trial. We have CT on the first row. F-MISO-PET on rows 2 and 3 using a couple of different metrics, tissue to blood ratio. I'll ask you to focus on the second row here. So here we can see our solitary tumor. We see a baseline level of hypoxia. After 18 gray, 48 hours later, we actually observed an increased tracer binding in our tumor, and then 4 days later, a decrease back towards baseline. So let's put some numbers on this. We've had 6 patients enrolled in this protocol so far. This was the first one that I showed in the previous slide. After 18 gray, 48 hours later, we actually saw a doubling of the hypoxic fraction in this patient. So we basically induced hypoxia and increased the absolute volume in this patient. That was not true for all of our patients. So the next two, actually no observable hypoxia at baseline or in response to 18 gray. And then patients 5 and 6 showed a similar trend to patient 2. So there's this is preliminary data, but there's a couple of interesting points that we're beginning to take away from this. There is potential for large variation in hypoxic fraction post-SBRT. And there is certainly heterogeneity between baseline levels of hypoxia between patients. It's quite significant, and I think that's also consistent with our classical understanding. And I believe that this does provide opportunity for therapeutic intervention, not only for patient treatment individualization, but also for potential to play with the timing. You know, is 48 hours really the best separation between our fractions, or maybe we should look at something longer, like 4 days or a week. Let's talk a little bit about clinical data for the last few minutes. We performed a meta-analysis last year that was published in the Green Journal with David Brenner at Columbia, looking at local control data for early stage non-small cell lung cancer and brain metastases. So on the left, we've got the non-small cell, on the right, the brain mets. And we collected data from the literature over the past 15 years reporting tumor control probability at greater than 1 year, the fraction number, and the dose. And there were about 33 studies with nearly 3,000 patients using 59 dose regimens, about a third of those being single fraction. So here we can see in both plots, the green is the single fraction data, the blue squares is the multi-fraction data. The black line here is sort of our standard, most basic TCP formalism with a linear quadratic model. And the red line is a TCP formalism that includes a distribution of radiosensitivity parameters because we know the heterogeneity in radiosensitivity in our human tumor populations. So you can see that the heterogeneous TCP model is less steep and does seem to fit the data more realistically. There's a few kind of tentative conclusions that we can draw from this, in my view. We observe a monotonic increase in tumor control with BED. So if we correct our dose prescriptions for this fractionation effect, we actually see as we deliver higher BED, whether it's single or multi-fraction, we see a corresponding increase in tumor control. And I believe as we deliver higher tumor BEDs, we're seeing higher levels of local control, and this may simply not have been practical with our conventional radiotherapy techniques. I'll also talk a little bit about high-dose models. So the question has been raised in recent years, is the LQ really an appropriate model at stereotactic doses? And we know that it's only an approximation to more sophisticated reaction rate models. When you get high dose rates, high doses, you tend to see an over prediction, whether that's at 5 to 10 gray, sort of has been the range where people begin to question it. Above 15 gray, you see this over prediction. And of course, this is neglecting any potential secondary biological mechanisms. So the question, again, we tried to ask in this paper, can we get a better fit to this clinical data using any of these alternate high-dose models than the standard approach, the LQ with heterogeneity in it. There's been a lot of in vitro studies looking at this question, but none really trying to ask this with clinical data. So we see that same plot that I showed in the previous slide with the LQ model, and then we see the linear quadratic linear model here, the PADE linear quadratic model, the universal survival model. There's probably a handful of others that we actually included in our analysis, and only 3 that I'm showing here. But the conclusions that we drew from this analysis were that the clinical data were most consistent with predictions of the LQ model with this heterogeneity and radiosensitivity over the whole dose and BED range. And we did not find that the addition of extra high dose terms really provided better agreement compared to the LQ with heterogeneity. And we did a statistical testing of this using a Kaike information criteria, which actually looked at the complexity of the model and how much better you could fit the data set, because you could always fit a data set better by adding additional parameters. So my last slide, and I can see that I'm already going over, what about the question of single fraction versus multi-fraction? So there's also a lot of debate around this. And this is the BrainMet data. And of course, all these conclusions looking at the clinical data have to be very careful, because there's a lot of heterogeneity when you're looking at multi-institutional data. But what we could draw from this, looking at the multi-fraction, the blue versus the single fraction, was that for brain metastases, the analysis suggested that multiple fractions may have a slightly higher effectiveness than single fractions, and at the very least, there was no clear evidence that single fractions were more effective than multiple fractionations. And we believe that this sort of is consistent with our, our classical understanding. In the context of hypoxia and reoxygenation, we would expect multiple fractions to do slightly better. And I think if we actually start to look more carefully at the hypoxic information of our tumors, we may be able to reduce some of this heterogeneity in the data. So in conclusion, there's a few points. I believe we still need to be cautious with extreme hypofractionation due to the effects of hypoxia. And I think, at least for early stage non-small cell lung cancer and brain metastases data, the available local control data don't really provide any clear evidence that we really need to invoke these secondary mechanisms to explain the excellent clinical results we're seeing. But I'm not going to just dismiss all this excellent in vitro and laboratory work. I think the effects are real. My question is whether they're secondary to sort of our primary effects. I think the LQ model does provide a reasonable approximation at stereotactic doses. And of course, clinical data is the gold standard, so we need to continue to test these hypotheses with better, more homogeneous clinical data sets. So with that, I'll end, and I thank you for your time, and I welcome any questions.